Hello, my name is Barbara Novak, and today I will be talking about my poster, Breaking Computational Barriers, NASA's Standardized Approach to Processing and Sharing Space Biology Omics Data. NASA's Open Science Data Repository, or OSDR, comprises three different NASA projects. The Ames Life Sciences Data Archive, ALSDA, which is the official repository for non-human data generated by NASA's Space Biology program at Ames Research Center. GeneLab, a repository for omics data from spaceflight and corresponding analog experiments, and the NASA Biological Institutional Scientific Collection, or NBISC, a biorepository of non-human samples collected from NASA-funded spaceflight investigations and correlative ground studies, which has recently been joined by the Space Microbial Culture Collection, or SMCC. Our mission is to enable the use of multimodal and multi-hierarchical fundamental space life science data to advance basic science, applied science, and operational outcomes for space exploration and knowledge discovery. OSDR provides a single submission portal, research data submission agreements, and workspace for all of this data, with an emphasis on maximally open access with necessary controls for sensitive data. We strive to keep our data maximally fair, findable, accessible, interoperable, and reusable, as well as transparent, inclusive, and reproducible. To fulfill the FAIR principles of open science, OSDR works with the scientific community via analysis working groups to help define both metadata standards and develop consensus data processing pipelines, which are used to generate and publish process data. The AWGs also collaborate to mine, reuse, OSDR data for new scientific analysis. Currently, there are 9 AWGs with over 1,000 members. If you are interested in space biology and would like to learn more about joining our AWGs, please visit nasa.gov/OSDR Open Science Analysis Working Groups. To make data interoperable and accessible, we first need to make sure that the metadata is structured and standardized. To accomplish that, we adopted the ISATAB format, consisting of an investigation file, a sample file, and an assay file. All of this metadata points to downloadable data files that are also use standardized formats. Metadata templates containing our standard ontologies and metadata structure are available on GitHub in the NASA/OSDR data curation repository, which you can see in the screenshot on the left. The screenshot on the right shows a typical sample and assay table from a study page in the OSDR web portal. In order to make OSDR data and biospecimens findable and reusable, we have created several interactive search and visualization tools. A data repository web portal provides a view of each study and simple filters and search options. For more complex queries, users can access all data in OSDR via the biological data APIs, which have both a REST and query interface for easy and powerful access. OSDR can also be explored visually via our friendly biological visualization shuttle, which can also pull together data from multiple studies. NBISC specimens are searchable via the NBISC biospecimen search tool. Finally, our environmental data application and RadLab portals provide specialized visualizations for environmental and radiation data, respectively. Another aspect of accessibility is making sure that users of all skill types can access it. For complex data types, such as sequencing data, it can be difficult for biologists without computational background or access to compute resources to make use of the raw data. For these types of data, we work with our AWGs to build standardized pipelines and workflows to process the raw data into a final processed format that users can access on their own laptops, and then host that data in OSDR as well. Our pipeline descriptions are designed to provide enough detailed information to allow computational biologists to replicate how we process data at GeneLab. The highlighted image depicts the bulk RNA-seq consensus pipeline. Our pipeline documentation is all hosted at our GitHub landing page at NASA GeneLab Data Processing. We use our consensus pipelines to build functional next flow workflows, which we use internally to generate standardized processed omics data hosted on OSDR. Our workflows are hosted alongside the pipelines in our GitHub repository, where they are documented with detailed installation and usage instructions. As you can see in the image, our workflows incorporate our consensus pipelines, but also add implementation of validation and verification of the process data along the way, the purple section at the bottom of the image. They also use containers or packaged environments to ensure reproducibility. We have developed a set of on-demand bioinformatics training courses to help biologists understand our pipelines and gain familiarity with basic bioinformatics tasks. Please visit my colleague Amanda's talk titled Gene Lab for Colleges and Universities, GL4U, on-demand bioinformatics training using space biology omics data on Wednesday, July 23rd in room 11A at 1520 BST. Now that I have talked about how OSDR is structured, I'd like to share some numbers. We currently host 555 studies, 1,023 datasets, over 45 species, more than 90 assays, more than 261 terabytes of data. In the pie charts, you can see how this data breaks down into different assay types and organisms. There are currently 117 original publications linked to OSDR, and we add to these constantly by pulling in data from NASA-funded experiments, as well as data hosted in other data repositories, such as SRA. There are also 124 enabled publications, that is, publications from analysis performed on data that was pulled from OSDR. Those enabled publications cover over 170 different datasets. Over time, the total number of enabled publications has started to grow faster than the number of original publications, showing that scientists have been able to perform new research with our data. The last piece of the puzzle is enabling users to easily analyze their own data using our standardized pipelines, even if they don't have access to their own compute resources. To that end, we have started a collaboration with Los Alamos National Labs, in which we are integrating the GeneLab Nextflow workflows into the NASA Edge Bioinformatics platform, which is equipped with access compute resources. You can see our first hosted workflow for processing Illumina amplicon sequencing data at nasa.edgebioinformatics.org. I would like to thank all of the members of the Open Science for Life in Space teams and our AWGs. And I would also like to thank the NASA Space Biology Program, NASA Biological and Physical Sciences, NASA Science Mission Directorate, and NASA Human Research Programs for their support. Thank you very much.